Hello dear students. Today we are going to start a new session describing the chapter Molecular Basis of Inheritance. This chapter is divided into 3 sections. Now we are discussing the first session of Molecular Basis of Inheritance. Okay. Now we can discuss the differences between the 2 types of nucleic acids, namely ribonucleic acid or RNA, deoxyribonucleic acid or DNA. First one, RNA. RNA is a single-stranded molecule. Single-stranded means it consists of one chain. That means like this. A single-stranded molecule. But in the case of DNA, 2 parallel strands are there. That means in the case of DNA, you can see that this is one chain and this is another chain, and these 2 chains are interconnected. That is why the RNA is called SS and the DNA is called DS. SS means single-stranded, DS means double-stranded. Another difference is that the structural units are, in the case of RNA, RNA is made by the association of large numbers of units, and these building blocks are called ribonucleotides. In the case of DNA also, nucleotides are present, but the nucleotides are called deoxyribonucleotides. So both RNA and DNA are made up of building blocks called nucleotides. But in the case of RNA, the nucleotide variety is called ribonucleotide. The DNA, the nucleotide variety is called deoxyribonucleotide. And this difference is based on the type of sugar present. In the case of ribonucleotide, the sugar is ribose type. In the case of deoxyribonucleotides, the sugar will be of deoxyribose type. Okay. Then, in the case of RNA, the base pairing is absent. That means the nitrogenous bases of one strand pairs with the nitrogenous bases of adjacent strand. In the case of RNA, as you know that there is a single strand, there is no pairing. But in the case of DNA, since 2 strands are there, the nitrogenous base of one strand will be pairing with the nitrogenous base of adjacent strand. Another difference is that in the case of RNA, the nitrogenous bases are adenine, guanine, cytosine and uracil. But in the case of DNA, the nitrogenous bases are adenine, guanine, cytosine and thymine. Instead of uracil, there will be thymine. So here adenine pairs with the thymine, guanine pairs with the cytosine, cytosine pairs with the guanine, thymine pairs with the adenine. That is the base pairing. So here the pyrimidine bases are cytosine and uracil. Here the adenine and guanine are purine bases. Cytosine and uracil are pyrimidine bases. Okay. So here also in the case of DNA, adenine and guanine are purine bases, cytosine and thymine are pyrimidine bases. So in both RNA and DNA, adenine and guanine represent the purine bases. Then the pyrimidine in the case of adenine, in the case of RNA, cytosine and uracil are the pyrimidine bases. In the case of DNA, cytosine and thymine are the pyrimidine bases. Okay. In the case of RNA, the sugar is ribose. The ribose, the second carbon of ribose has OH group. So the deoxyribose sugar, the second position of carbon has one oxygen less, only hydrogen group will be there. Since RNA is a single-stranded molecule, it is structurally less stable as compared to DNA, and therefore the mutation rate may be very high. But in the case of DNA, 2 strands are connected together. Moreover, they are formed in the form of a double helix, so that they are structurally more stable and the mutation rate is comparatively low. Then regarding the function, the RNA contains the code for the synthesis of protein by a process called translation. And the DNA is involved in the production of RNA by another process. That process is called transcription. So from DNA, RNA can be produced by transcription. From RNA, protein can be produced by translation. Then, how the RNA is produced? RNA is produced by transcription from the DNA. And how the DNA is produced? DNA is produced by replication from a previous DNA. A mother DNA replicates to producing daughter DNA, and the RNA molecules are transcribed from the DNA template. Then the RNA molecule will be acting as genetic material in some retroviruses only. But in the case of DNA, in all other organisms, DNA is the main genetic material. So these are the basic differences between two types of nucleic acids, namely RNA ribonucleic acid and DNA deoxyribonucleic acid. The next prime point is regarding the nucleotides. As you know, in the case of both RNA and DNA, the building blocks are called nucleotides. Okay? So the nucleic acids are produced by the structural units called nucleotides, and each nucleotide comprises of three parts. There is a sugar unit, this is the sugar unit. It's a pentose sugar, 5-sided ring structure, and this will be carbon 1, this will be carbon 2, this will be carbon 3, this will be carbon 4. And the carbon 5, the fifth carbon atom, remains outside the ring structure. So this is the ribose sugar. Then a nitrogenous base that can be either purine or pyrimidine is attached with the first carbon atom of the sugar. The nitrogenous base combines with the first carbon atom of the sugar by using a glycosidic bond. The combination of nitrogen base and sugar, this unit is called nucleoside. This is nucleoside. Nucleoside, in short, we can write as NS, nucleoside, N representing nitrogenous base, S representing sugar. Then, attached to the fifth carbon atom, a phosphate group may be linked. Now the nucleoside becomes nucleotide. The phosphate is attached to the fifth carbon atom by means of an ester bond. Ester bond. So, a pentose sugar, attached to the first carbon atom there is a nitrogenous base, attached to the fifth carbon atom there is a phosphate unit. So this combination is called nucleotide. So they are the nucleotides are the structural units of nucleic acids. Each nucleotide consists of a phosphate group, a pentose sugar. The pentose sugar will be ribose in RNA and deoxyribose in DNA. So ribose means in the attached to C2, that means carbon 2, the second carbon, there will be CHOH. That is called ribose. If this oxygen is absent, it is called deoxyribose. Then there is a nitrogenous base. The ribose sugar is present only in RNA, deoxyribose sugar is present only in DNA. So three units are there, a phosphate group is there, a nitrogenous base is there, a sugar molecule is there. Then the nitrogenous bases can be purines or pyrimidine. The purines are adenine and guanine, both in RNA and DNA. The pyrimidines include cytosine and uracil in RNA, cytosine and thymine in DNA. So two types of purine bases and three types of pyrimidine bases. A nitrogenous base plus pentose group, pentose sugar, that means that unit is called a nucleoside. Nitrogenous base plus sugar. When the nucleoside combines with the phosphate group, that combination is called nucleotide. Then, two nucleotides, the adjacent nucleotides in an RNA or DNA are linked together by 3,5-phosphodiester linkages. That means a diester linkage is produced between fifth carbon atom of one sugar and the third carbon atom of adjacent sugar, mediating a phosphate group. So that bond is called phosphodiester bond. Okay, this is the organization of a nucleotide. Nucleotides are the building blocks of nucleic acids. Okay. Next prime point is regarding the nucleosome. Nucleosome is a structure associated with package of DNA double helix. So here the DNA double helix molecules are packaged in the form of structures called nucleosomes. So here the DNA molecule is a negatively charged molecule because DNA has phosphoric acid and the acid donates a proton so that the residue becomes negatively charged. and that will be wrapped around a positively charged histone protein to form a nucleosome. Suppose this is a histone protein. It is a positively charged protein where basic amino acids such as lysine and arginine are abundantly present. And the DNA molecule, which is negatively charged, is coiled around the histone protein. The histone plus the wrapped DNA together form what is called a nucleosome. So histones are rich in lysine and arginine are organized to form a unit of 8 molecules. The histones are comprising of 8 units. So the histone is always called histone octamer. Okay, suppose this is a histone octamer. 8 units is surrounded by the DNA coils. One nucleosome has around 200 base pairs of DNA helix. The total length will be 200 base pairs. The adjacent nucleosomes are interconnected by linker DNA. Suppose this is another histone protein, again forms the nucleosome. So this is another nucleosome. Adjacent nucleosomes are interconnected by a DNA. That DNA is called linker DNA. Long chains of nucleosome constitute chromatin. You can see that there is very long chains of nucleosomes, just like beads on zinc structure. And this is what is called chromatin. Then the chromatin molecules are again packed together by non-histone proteins so as to form thickened bodies. The thickened bodies are called chromosomes. So in order to make the DNA molecule compact, it is coiled around the protein molecules called histone, forming chromatin, and the chromatins are again coiled together by non-histone proteins, forming what is called chromosomes. This is organization of nucleosome. Now we are moving on to another very important principle in molecular biology, that is called central dogma. Central dogma is a central idea. The central dogma refers to the pattern or flow of information which form the basis of all metabolic functions of the body. That means information contained in one molecule is transferred to another molecule by a sequence of events. So here, according to central dogma, the information is primarily present in DNA. All the information for the functioning of cell is primarily recorded in DNA. And one DNA can produce another DNA. One DNA can produce its copy by a process called replication. That means when a cell divides, or DNA can be multiplied or replicated to produce another DNA having the same information. So information transfer from DNA to DNA is called replication. Then the information can be transferred from DNA to RNA. So that kind of information transfer is called transcription. It is called a transcription. Then the RNA, which is receiving information from DNA, will pass on its information for the synthesis of a protein by another process. That process is called translation. So information transfer from DNA to RNA is called transcription. RNA to protein is called translation. So as per central dogma, information transfer from DNA to DNA is replication. DNA to RNA is transcription. RNA to protein is called translation. Then in some retroviruses, the reversal is taking place. That means from RNA, a DNA molecule can be synthesized. That means information transfer from RNA to DNA. So that process is called reverse transcription. Reverse transcription were first of all described by Temin and Baltimore, so that the central dogma reverse is otherwise called Teminism. The first one is called central dogma. The reverse process is called central dogma reverse. The central dogma was described by Francis Crick. In central dogma reverse, informational transfer from RNA to DNA, the process is called reverse transcription. So here the central dogma represents the basic idea of informational transfer from one system to another system, DNA to DNA, DNA to RNA, RNA to protein. Okay. The next prime point is regarding RNA world. RNA, being structurally similar, was the first genetic material. All the life processes were initially evolved around RNA molecule. In the case of some viruses, the RNA was the genetic material. And also RNA is working as a catalyst, like enzymes. The RNA enzyme is called a ribozyme. Since RNA being catalyst, it is reactive and hence unstable. So it mutates at a higher rate. More stable genetic material was evolved from RNA, that was DNA. Since DNA is DS means what? Double-stranded. It is more stable than RNA which is SS. But anyway, initially the RNA was the first genetic material. So that concept is called RNA world. Okay. The next prime point is replication. It's a very important point. What is replication means? One DNA from a mother DNA, two daughter DNA molecules are produced. The synthesis of daughter DNA from mother DNA is called replication. The replication occurs in such a way that there is no change in the genetic information. That means that both the daughter DNA produced will be identical to each other, and these daughter DNA will be identical to the mother DNA in their genetic information. Okay. So how the process takes place? The synthesis of daughter DNA strands from mother DNA without losing information contained in them is called replication. And this is mediated by so many enzymes, and the main enzyme is called DNA-dependent DNA polymerase. The synthesis of DNA is by DNA polymerase, and the DNA polymerase is working on the basis of another DNA. So the enzyme is called DNA-dependent DNA polymerase. So what happens during replication? Only the main events. So the DNA molecule is double-stranded, isn't it? It is a double-stranded molecule. So at the time of replication, the two strands will separate from each other and the base pairs will be broken. Base pairs will be broken, and both of the separated strands will act as a template. Template means a master copy for the synthesis of daughter DNA. So based on one template, one daughter strand is produced. Based on another template, another daughter strand is produced. Okay, this is the daughter strand produced, and here this will be the daughter strand produced. So this is the mother strand, this is the mother strand, and these are the daughter strands. So in the daughter DNA, you can see that this is a daughter DNA. In the daughter DNA, out of the two strands, one will be taken from the mother and only one will be newly synthesized. The second daughter DNA also, one will be taken as such from the mother and the other will be newly synthesized. That means in each daughter DNA, one strand is mother's DNA and the other strand is newly synthesized. That means the replication pattern is called semi-conservative method. You can see the two strands of the DNA will separate and act as a template for the synthesis of new daughter strands. After the completion of replication, each daughter DNA produced will have one parental strand and one newly formed strand. That is why the scheme of replication is called semi-conservative method. Okay. The next prime point is transcription. We already studied that transcription means a synthesis of RNA from a DNA molecule. The information from DNA to RNA is called transcription. DNA, you know that DNA is a DS molecule, RNA is an SS molecule, single-stranded. So here, during the time of transcription, this is the double-stranded DNA molecule and it separates from each other. The two strands will separate at a particular point. Okay. And one will be acting as a template and another never enters into the formation of RNA. And that strand which never enter into the formation of RNA is called a coding strand. Based on the template strand, an RNA strand is produced. This is the RNA strand produced. Okay. So the RNA produced will be complementary to the template. The template is already complementary to the coding strand, so that the RNA produced will be structurally equal to the coding strand. You can see here the transcription refers to the copying of genetic information from one strand of DNA into RNA, as per the principle of complementarity. The main enzyme for transcription. So here the process is the synthesis of RNA, so the enzyme is called RNA polymerase. And the RNA polymerase work on the basis of a DNA, so it is called DNA dependent RNA polymerase. So here the transcription process is initiated at a point. The process is called initiation, and for the initiation a factor is needed. That factor is called sigma factor. So sigma factor is needed for initiation. Then the process will be continuing through the template, and the process is called elongation. For elongation, no additional factors are needed, only RNA polymerase will be working for elongation. And once the RNA synthesis is completed, the process is called termination. For termination also another factor is needed. That factor is called rho factor. So sigma factor is the factor for initiation, rho factor is the factor for termination. So the RNA synthesis, that means transcription, can be comprised of three events: initiation, elongation and termination. Okay. The next prime point is transcription unit. We already studied the mechanism of transcription, and the transcription take place at a particular region, and that region is called transcription unit. It refers to the actual region where transcription take place. So in the transcription unit, you can see there is one DNA strand acting as a template. This is a template strand. Okay. This is a separated DNA strand. Its complementary strand is here, and the complementary strand is not taking part in the process of transcription, and that is called a coding strand. So template is a single-stranded DNA, coding is also a single-stranded DNA. And the template strand, the direction, the orientation will be 3', 5'. So the coding orientation will be 5', 3'. A new RNA strand is synthesized. A new RNA strand is synthesized based on the template strand. This is the RNA. This is the RNA produced. So the RNA synthesis takes place in 5', 3' direction. The synthesis always take place in 5', 3' direction. The RNA that's produced is called a transcript. The product of transcription is called a transcript. So template is DNA, transcript is RNA, coding is also DNA. So this is the starting point, and this is the ending point. The starting at the 5' end of the RNA, stopping at the 3' end of the RNA. That means starting at the 3' end of the template, stopping at the 5' end of the template. The starting region is called promoter site. This is the promoter region. The ending region is called terminator site. And the part of the template DNA in between the promoter and terminator is called structural gene. It is called structural gene. So here the two strands of DNA in the transcription units are denoted as template strand, which is 3', 5' in orientation, and coding strand, which is 5', 3' in orientation. It comprises of three essential regions such as promoter region, structural gene and terminator region. What is promoter region? Promoter is the starting point of transcription located at the 3' end of the template. Terminator is the ending point of transcription and located at the 5' end of the template. Structural gene is seen in between promoter and terminator region. So this is the orientation of transcription unit. Transcription unit means the region where actually the process of transcription, the process of RNA synthesis take place. Okay. The next prime point is regarding the types of RNA. There are three major types of RNA such as messenger RNA or mRNA, transfer RNA or tRNA, ribosomal RNA or rRNA. The messenger RNA acts as a template for protein synthesis. Transfer RNA is used for transporting the amino acids towards the site of a protein synthesis. The ribosomal RNA plays a structural and catalytic role during the process of translation. So in prokaryotic organisms, the three types of RNA, that means mRNA, tRNA and rRNA, are synthesized by the single enzyme called DNA-dependent RNA polymerase. But in eukaryotes, for each RNA, specific RNA polymerase enzyme is there. You can see that in the RNA polymerase 1 is for the synthesis of rRNA, RNA polymerase 2 is for the synthesis of mRNA, and RNA polymerase 3 is used for the synthesis of tRNA. Okay, these are the three different types of RNA polymerases working for the three different types of RNA synthesis. By this, we are ending the first session of molecular basis of inheritance. Thank you.